very pleased to welcome Professor Alan Trounson. He's President of the California Institute for Regenerative Medicine. During his career, Professor Trounson led a team to discover human embryonic stem cells, directed their differentiation into nerve and other tissue cells. He was a director of Victoria's Monash Centre for Early Human Development before becoming the founding Deputy Director and Director of the Institute for Reproductive Biology. From there, he created the first Australian Biotechnology Centre of Excellence, the Australian Stem Cell Centre, and also went on to establish the Monash Immunology and Stem Cell Laboratories before becoming President of the California Institute for Regenerative Medicine, where he's overseen extraordinary development, leading to more than 1,000 publications. He currently holds an ambassadorial role at Veski and was a founding director and former Veski chairman before relocating to San Francisco. Alan Trounson.
Thank you, Jonathan. It's a pleasure to be here. A real pleasure to have just got off the plane today from San Francisco. And I'm very happy that Julia, who's been with Veski right from the beginning, has chaperoned what I think has been a tremendous organisation. And my friend Snow Barlow, who's been chairman of Veski for the last 6 years, I think, Snow, has really turned this into something that's incredibly valuable. Well, what about healthy ageing? Australian science and medicine is undoubtedly first rate. And just to remind you, there are plenty of scientists with a great record in international science in the medical community. Their Nobel laureates feature regularly, from Brian Schmidt in physics in 2011 to a raft of laureates in physiology and medicine, including Elizabeth Blackburn in 2009 for physiology and medicine. For the chromosome protection by telomeres and the enzyme telomerase that extends the multiplication of cells. Barry Marshall and Robert Warren in 2005 for the discovery of the role of Helicobacter pylori in gastric ulcer disease. Peter Doherty in 1996 for cell-mediated immune defence. Bernard Katz in 1970 for humoral transmitters in nerve terminals. John Eccles in 1963 for ionic mechanisms of nerve cell membranes. McFarlane Burnett in 1960 for acquired immune tolerance. And Howard Florey in 1945 for penicillin. There are many other great medical discoveries that have arisen recently from Australian laboratories, including Ian Fraser's HPV vaccine for preventing cervical cancer, Don Metcalf's colony-stimulating factors that have impacted medicine very broadly, Carl Wood's work on human IVF, among many others. One might have cause to be optimistic about the next 20 years, maybe even expect Australia to have a Minister of Science driving high-tech developments and a strong biotechnology industry employing scientists and medical graduates maturing in universities and research centres. This might rival the quantum benefits of the mining boom and particular sectors of the food industry that appear to rise and fall pretty regularly. However, it doesn't take much searching to demonstrate the disconnect between science discoveries and community utilisation of the potential discoveries. What is hampering our smartness in converting high-quality science discoveries to community benefit? Is Australian science research sustainable without the ability to expand beyond the publicly funded institutional base of universities, medical centres and CSIRO? Where's our biotech industry? Who spans the so-called valley of death? There are distinct scarcities of industry connected with the Australian medical research community and a lack of venture investment in the biosciences. However, companies such as Mesoblast can arise in the Australian environment to be the world leader in stem cell medicine, with a market cap of more than $10.4 billion. And Silvio's a smart guy, but he's a lonesome guy out there in an area where we ought to have an incredible growth. In California, I developed academic research teams partnered with companies. or utilising commercial consulting together with independent and in-house guidance to successfully navigate the translational pathway to clinical trials. Beginning in 2010, from pure discovery base, our so-called disease teams are now entering registered clinical trials. Expect 10 by the end of 2014. They're now connected to industry and expected to fare extremely well. Areas such as the destruction of malignant cancer cells, cure of genetic disease such as beta thalassemia and sickle cell disease, cure of HIV/AIDS, cure of blindness, particularly macular degeneration and retinitis pigmentosa, cure juvenile type 1 diabetes, and treatment for heart muscle damage from myocardial infarct, heart attack, spinal cord injury, and the deadly motor neuron diseases. Well, what about a big idea for 2030? And I'm going to give you one in immunology and stem cells, defeating cancer. In 2030, we might expect the research has delivered us a way to defeat cancer by engineering the T-cell receptor, TCR it's called, to recognise cancer-specific antigens. These are the proteins that are specifically expressed by cancer cells. This chimeric antigen receptor, or CAR, C-A-R, CAR technology, could be introduced into patients' T cells. The T cells are the scavenging killer thymic instructor cells that aggressively target foreign infection or abnormal or non-self cells. So they're the really aggressive part of our immune survival. So we could target them directly or target them through blood or pluripotential stem cells. The TCR, the T cell receptor, is expressed only in T cells and is activated to destroy malignant cancer cells expressing any abnormal antigen of the patient's tumour. We can select these cells for expansion or genetically engineer them to be extremely potent killers of our cancers. Now we have an internal surveillance system capable of removing any cancer stem cell that develops. This can last a lifetime if introduced through the stem cells in the body, or be made patient-specific in large numbers by converting the patient's own T cells to what we call iPS cells, induced pluripotential stem cells, the cells that we can take from anyone now and turn into something that's equivalent to an embryonic stem cell. So if you take a patient's T cells, convert them to iPS cells, and then grow them back out to T cells, they'll express the cancer-specific antigen, and you can use those for cancer therapy. And if you make iPS cells, we can make billions and billions of those. So we have a very effective therapy for your cancer. So we'll expand them and redirect them into the activated T cells for tumour-killing purposes, and this would be an effective T cell therapy. The beauty of this system is it can be modified and tailored to work in concert with an active immune system, not against it. So usually in cancer therapy, you'll recognise we give the patients heavy immune suppression because we want to use therapies to down the immune system. Well, this is not this system. We should also be able to reawaken the immune system for recognised and tolerating autoimmunity. The thymus can be regenerated now by cell therapy or with small molecules. So the thymus is the organ that's the last discovered organ in the body, which is responsible for instructing the T cells about self and non-self. It's a critical organ in the body, but once you go beyond puberty, it starts to regress. So it goes from an orange in the size of an orange in a 1 or 2 year old child to a tiny shriveled up peanut, or worse in a person of my age. So they've figured out how to reactivate that, and you can use small molecule, for example, targeting thymic epithelium transcription factor called forkhead box N1, or FROXN1, by Blackburn and colleagues in Edinburgh, and just published just recently. Wow, very Melbourne. Foreign or allergenic cells and tissues will be tolerated, and potentially universal donor tissues will be available for immune-compatible use by patients. This requires we apply the power of tools discovered in immunology together with developments in stem cell biology and ensure we translate for the patient benefit. And Melbourne is the capital, the world capital for immunology, and it was and should be the capital for stem cells. At least Silvio is a beacon for stem cells in the Mesoblast company here in Melbourne. What do we need? It's time that academics of science, funding agencies and government looked at such models as to refocus the limited public resources available in a much more strategic way. We need to really encourage the very smart academics to become interested in applying their discoveries for the community benefit. Universities have to change how they recognise and reward scientists who enter this pathway, not penalise them. It isn't about the number of publications or grants from the NH and MRC. These are not the measure for making the innovative contributions needed. They probably handicap the conversion to a more effective system that the community will recognise and endorse. The current federal grant system is broken in the USA. You can read about that, Albers and colleagues in PNAS this year. And would not be any better here. Probably worse. Money is trickling down an unsustainable pipeline that fails to encourage developments for use by the community. The system must be fixed despite the inertia that exists even amongst the majority of scientists. We'll have no viable research environment soon if we continue to look away from the problems that are evident and are turning away our smart young scientists. We must quickly revise the direction we're taking. And one of those very smart young scientists is one of my sons, who said he didn't want to spend 6 to 8 months of every year applying for research grants that he didn't get. An incredibly smart guy, but he's not going to go into science. And many of the people that he worked with in the Walter Eliza Hall had the same view. These are young people who are being turned off. We need a viable and robust biotechnology industry with investors who are confident in the returns on investment. Australia is an attractive place for basic research, but it needs to become competitive in translation of these discoveries. Preclinical demonstration of safety and sound proof of concept for the candidate products with the accompanying regulatory requirements for pharmacokinetic data, processing, manufacture of the therapeutic candidate, development and validation of potency assays, etc., are areas of biotech experience that should be available for the raft of opportunities that exist here for translation. Typically, patent lawyers drafting patents should have experience in prosecution of IP. I think we need to link with US biotechnology industry to move the capacity, this capacity, into places in Australia where sufficient clinical and scientific power exists. Melbourne is one of those key places. The experience might be illuminating and probably rewarding. So get on with it and get global. The Australian Research Committee must be globally linked to the other big engines of progress. This isn't a feature of research in Australia. Indeed, it's rare for universities' researchers in the same city to collaborate. I worked at Monash, and I rarely, if ever, collaborated with anyone with Melbourne University. Indeed, we cannot be competitive by working in isolation. Niche discoveries aside, we're less represented in the major journals with time, less recognised and less respected. The Asian tiger research communities, China, Japan, Korea and Singapore, are moving strongly to challenge North American dominance. And Europe is awake to the merits of collaborating. They use vehicles such as EU funding and granting. I encouraged Australians to collaborate with California in stem cells. They've been extremely competitive. They were awarded more collaborative grants than any other country besides Germany. These high-quality scientists are currently starving for funding here in Australia. We need to re-evaluate the merits of becoming global partners in medical research and sharing the benefits with our collaborators that will flow from this work. We need to communicate with the whole community, inform them of the extraordinary opportunities of wealth and health creation. Time is of essence here. The so-called Australian clever country will not be recognised as that in the next decade or two without major change. We must change the way we fund research. We must consider how to reward scientists properly. We must expand the opportunities for expressing their smart ideas. We need to power up industry linkages to and partnerships with academia and medicine. We need to globalise our collaborations. We need to make science and medicine an industry to compete with resource-based commodities for wealth and prosperity of the whole Australian community. Most of all, we need to begin a dialogue with the Australian community about the benefits of medical research for our healthy ageing. I'll give you an example of returns over 23 years of $1,400 for every $1 invested in the original research grants for sequencing the human genome. $1,400 for every dollar invested. I think that would make everybody pleased in the private sector. And there's likely large economic benefit from investing in medical research in stem cells, pathogenic vaccines, cancer drugs, etc. And this needs to resonate in the halls of government, not just be a whisper from scientists returning to Australia with the aid of veski, or in my case, after managing $3 billion of Californian bond funds to drive a revolution in regenerative medicine. It might take 20 years to reward so well as this kind of investment, as it did in the human genome, but we need the support and we need the changes in the way we arrange this support for Australians to be living really smart and really healthy in 2030. Thank you very much.